Hello everyone, today we will be discussing glycolysis, which is the cellular energy journey from glucose to pyruvate.
Assalamualaikum and hi everyone.
My name is Aina.
My name is Naufal.
My name is Arvi.
And today we are going to be presenting about glycolysis.
This presentation is based on the Campbell biology. Let's take a look at the big picture. Glycolysis takes place in the cytoplasm of the cell. The input for this process is 1 molecule of glucose, a 6-carbon sugar. The output is 2 molecules of pyruvate, which are 3 carbon molecules. The entire process involves 10 enzymatic steps, divided into 2 phases. Phase 1 is the energy investment phase, and Phase 2 is the energy payoff phase. Phase 1 is from steps 1 to 5, and the energy payoff phase, which is step 2, is from step 6 to 10. Moving on to Phase 1, energy investment phase. In step 1, glucose is converted to glucose 6-phosphate and causes 1 ATP. In step 2, the molecule undergoes isomerization to become fructose 6-phosphate. In step 3, fructose 6-phosphate is converted to fructose 6-bisphosphate and causes another 1 ATP. The key takeaway here is that we have invested 2 ATP to prime the glucose for splitting. Here is a clear diagram to visualize the energy investment phase. Next, we reach step 4, also known as the great split. The enzyme aldolase splits fructose 1,6-bisphosphate into 2 different molecules, G3P and DHAP. In step 5, the enzyme triose phosphate isomerase immediately converts DHAP into G3P. The result is that we now have 2 molecules of G3P. It is important to remember that all following steps happen twice, one of each G3P molecule. Now we enter the energy payoff phase. In step 6, G3P is oxidized to form 1,3-bisphosphoglycerate. This reaction is catalyzed by G3P dehydrogenase and produces 2 NADH electron carriers. In step 7, phosphoglycerate kinase performs substrate level phosphorylation to convert 1,3-bisphosphoglycerate into 3-phosphoglycerate. This produces 2 ATP. At this point, we have broken even on our energy investment. Continuing with the payoff phase, in step 8, phosphoglycerate mutas locates the phosphate group, turning 3-phosphoglycerate into 2-phosphoglycerate. In step 9, the enzyme enolase causes a dehydration reaction, removing water to form phosphoenol pyruvate. Finally, in step 10, pyruvate kinase converts PEP into pyruvate via substrate level phosphorylation. This produces 2 more ATP. So, what is the bottom line for our ATP economics? We produced a total of 4 ATP, but we invested 2 ATP at the start. This gives us a net profit of positive 2 ATP. Additionally, we gain 2 NADH molecules for the electron transport chain and 2 pyruvate molecules for the Krebs cycle. To summarize, the net energy yield, the investment phase inputs 2 ATP, the payoff phase outputs 4 ATP and 2 NADH. The final net gain is 2 ATP, 2 NADH, and our final products are 2 pyruvate molecules. Here is an overview of glycolysis in the form of my math to give a short brief introduction to the topic. So now, what is going to happen next? If aerobic conditions are present, the cell moves to the Krebs cycle and electron transport chain. If anaerobic, it undergoes fermentation. Glycolysis is ancient, universal, and essential. That will be all from us. I hope the video can be a bit of help for you. Thank you.